that is fairly destructive to habitats like this. So it's something that's one of the reasons why we survey these areas, so we understand what's there, and we can make recommendations for how these areas can be protected.
Thank you.
One of the regulations for the National Marine Sanctuary is no bottom disturbance, with the exception of lawful fishing practices. And that's because the fisheries management has authority over fisheries for the whole region. But that's why this science is so important, so we can contribute what we know about these areas to the decision makers about fisheries. And bottom trawling does have bottom disturbance, so it's a particular interest.
This is a nice distance from the substrate here. And we can keep moving along at this depth and see if we get over this little mud area and see if there's some more rock habitat up ahead.
Copy that. It looks like if I come up a little, we'll hit some rocks there.
Okay, let's do that.
Copy.
Yeah, I kind of like to be up on that contour where the ship is.
Yeah, so basically come north of where you are.
So I was checking out blob sculpin. They feed on sea pens, crabs, and mollusks.
Sea pens, really?
Mm-hmm.
I did not know that.
And then being a marine sanctuary, is this area protected from offshore drilling? One of our listeners wants to know.
Yes. That is one of the main reasons this area was included in the National Marine Sanctuary, because residents on the coast wanted that prohibition extended all the way up north. And one of the ways it connects is that the waters that we have upwelled along the coast, just north of us is some of the peak upwelling values that we get. And that upwelling, the water that's upwelled up here moves down the coast with the California current moving south. And as it moves, all those nutrients get utilized by phytoplankton, and the phytoplankton develop and bloom as they move south into the Greater Farallones region just outside San Francisco. And so that interconnectivity of those source waters and the productivity of where the food web really blooms and takes off is one of the reasons that ecologically it makes sense too. So we're very happy to have that. And this is a beautiful area that we just happened upon here.
There's a squat lobster inside this peristinella. So we've got some more of these peristinellas. We've got some nice images of them and a sample. So we can zoom out and keep going.
The background was really nice with all that cluster that we're just coming up on.
Yeah.
And those are Brisingid stars on the rock?
Yes.
Brisingids.
We see a lot of echinoderms on these dives, lots of different types of stars, including brittle stars, serpent stars, Brisingids, and sea stars. There's quite...
Interestingly, we're not seeing too many brittle stars in the sediment, like we saw earlier.
This is a really
Maybe
nice
they're hiding
outcropping here with all this Calagorgia, Brisingids, crinoids.
There's some brittles on the rock.
Yeah.
Yeah, this one has a a good sampling.
Wow, great
Okay,
diversity.
coming wide. Full wide.
You can see why we target these rocky areas, why that mapping data is so essential to exploration. Otherwise, we'd just be wandering around. Grenadier. Yes. Getting it.
Did he just?
Was he just feeding off of something on the
I
coral?
think he did. We'll have to review the tape, see if he got a brittle star or something out of there.
So wondering I have is, why is this spot such a hot spot for corals? We're seeing such a diversity here.
Yeah, as soon as we got to this rocky area, it was covered with corals.
Beautiful.
Yeah, this is the first time we're taking a look at this area at this depth in Greater Farallones National Marine Sanctuary. This area that was added to the sanctuary in 2015. So it looks like guys picked a good spot to add a sanctuary.
One of the reasons they requested boundaries to go further north and further west were to encompass deeper areas, because there are some slant drilling techniques that can come in from deeper waters. And so they wanted to go as wide and deep as possible to prevent that type of activity. And oil platforms wouldn't be very compatible for this region, for accidents happen all the time, and we have such precious ocean resources here, fisheries, beaches, huge part of the coastal culture and coastal economy. And our residents have said they don't want that here. It's nice that they're participating in these public processes to speak up for what they want to see in their federal marine protected areas.
You need to come north.
Great shot of the marine snow and the ctenophores floating by.
Look at this teeny little ridge. Sticking out.
Just a little bit of rock, and there's a bunch of stuff on it.
This is going to take us back east, though.
Someone tuning in was curious about the colors. Why are some of the colors of these corals and animals and anemones so
I
bright
think it's just that
when they
round
live
bit.
so deep and in such a dark environment?
That's a good question, and a lot of these animals can't see color, and other animals can't see them because it's so dark down here all the time. So some of the colors might be to help them stay camouflaged. Yeah, I'm not really sure.
The attenuation of red make red a very black down here.
Mm-hmm.
Oh, with bioluminescence, it would show up as dark.
Mm-hmm.
I'm wondering if these are some type of zoanthid covering this
Yeah,
rock.
let's take a look. Let's zoom in on that rock face. I was kind of wondering that myself.
Okay, zooming in on the moss.
Ooh.
So those are hydroids at the bottom, the frilly stuff, but on the rock itself are all these polyps. So there could be zoanthids. Yeah, I think that's what they are. Good eye, Jenny.
Okay, coming wide.
So plentiful. It's amazing.
Haven't seen many hydroids down here, so. Or have you been seeing them all along the way and I'm just not noticing them?
No, I haven't seen all that many. We have seen a few. I think we even got some along with some of our collections on the last dive, but yeah.
Full wide.
And hydroids are cnidarians? I'm asking, I don't know. Ooh. Hydrozoa. Some hydroids, such as the freshwater. Cnidarian order.
Oh, looks like we've got a Swiftia. Red coral up ahead. We'll take a zoom and some photos on that.
Go ahead, Wayne.
Which
Oh,
one?
and then another, the red coral. Just to the left of the lasers.
Got it. Yeah.
Nice. We collected one of these on our dive yesterday.
Uh-huh. All right, full wide.
Roger.
Yeah, I saw us processing it in the wet lab today.
Yeah, run the dust.
There's the
And there's an
octopus.
octopus.
Yeah.
So we're kind of going back the way we came, but at a different altitude. You guys are happy with that?
Back the way we came. We should be moving along this contour to the, what is that, the northwest towards the next, we can, I think we can skip over, if we follow along the contour, we'll be skipping over Guy 1 and going to the next waypoint.
Roger. We're kind of going to the northeast.
Yeah. So I think we're just going to have some mud in our future.
What's that?
We're just going to have some mud in our future.
What just happened?
So target bearing is 342.
Okay, hang on. I'm not sure we're on the same page. So if we follow this along this contour, we can skip Guy 1 and just follow the contour. Will take us up to Guy 2.
Correct.
So, okay, that's what we're doing?
That's what we should be doing, yes.
Yes, okay.
Okay.
So I have to...
I'm going to go 343, 342, 350.
We're about
Ghost line
1,700 meters down right now.
Hello, Mud. We've missed you.
I think
Antonella, when we're flying over the mud, can you tell us a little bit about piloting Argus? What are some of the variables that you're considering? And
yeah, so the sort of basic part of piloting Argus is it's attached with a long cable and a winch to the ship. And
There's
so
nothing out
you winch up and down to control Argus's depth. And you have to work with the Herc pilot a lot. Sometimes you need to come down farther to give them more leash so they can explore farther away. Sometimes you need to come up. And then you also work, well, the ship controls the position of Argus. And so wherever nav puts us, that's sort of where we can, like, Herc has a leash and can explore that immediate area. There are also 2 thrusters, which we can move laterally, but we don't usually do that. It's more controlling the heading.
What do you guys think about coming upslope a little bit to the next contour line? It looks like it might be a little steeper, and following that along.
Yeah, can do.
Sorry, Antonella.
No worries. Whoop. 1 sec. So oops, hang on. I just got pulled around.
Sorry, I'll
No worries.
slow down there.
So the other...
This is Guy. I think if you go from where you are right now to Guy 2, you're going to be in the mud. You might want to consider going upslope until you hit some rock and then go to Guy 2.
Got it, Guy. Okay, so we had just started moving upslope a little bit to see if we could get some rock, and Guy recommends that we do that and maybe even come upslope a little more directly to see if we can hit the rock and then turn along the contour line.
Copy. I am headed uphill.
While we're over the mud, if we see some eel pouts or grenadiers, we can get some stills on that.
all the way around.
Did I completely pull you around?
Yeah, I'm pretty much 180 from you.
Waiting on the ship.
If we're stopped, if we need to stop and get the crab and the eel pout there.
You want images of the crab?
Yeah, if we're stopping to wait.
We are stopped to wait.
Okay, well, let's get some photos.
So is that white color something that's typically seen on these crabs? I've noticed that on the last 2 or 3 crabs we've seen, the center of them being very pale.
Yeah, I think that's a species, a type of crab that has that. I gotta look that one up again.
Kind of looks like that Oregon hair crab, but that lighter carapace. I wonder if it's the same thing.
We're spinning.
I can back up a little.
Yeah, I'm not really sure it's able to fight.
A giant tumbleweed.
Huh? I'm gonna need to spin all the way back around.
As we kind of reposition ourselves, I just want to share a little bit from nautiluslive.org. There's a lot of really great resources on our website. So something that's pretty fun is to check out the team. You're able to click on different individuals, read their bios, learn about their career path, and hear some advice from them, which is really fun. If you're tuning in from a school, Under our education tab, we've got lots of great curriculum and lesson plans on different topics in the STEM, science, technology, engineering, and math that are definitely worth checking out. And then for clips and awesome videos from past expeditions, certainly go to photos and videos. You maybe noticed under the expedition tab that we have one more expedition after this one that we're on, which is we're currently in Cordell Bank National Marine Sanctuary and the Greater Fairlawns National Marine Sanctuary, but next we'll be going to Monterey Bay Marine Sanctuary and wrapping up our EV Nautilus season.
For some people, that's like a sad face right there.
I know.
I know we have viewers worldwide that watch all season long. But the good thing is all the replays are on YouTube, so you can go back and review.
Right. When we look up information on different species, we have a question from one of our viewers. They're curious what database we reference.
Well,
Okay, I'm back in position. Really?
well, in terms of species, I have been using a file a taxa ID guide that Caitlin actually made from our previous expeditions of species that we saw and identified to the level that we know for sure. And a lot of those have been cross-referenced with other field guides made by local researchers, Monterey Bay, Oregon. There aren't too many definitive deep sea guides for this region. It's pretty much as we go along, people continue to build their own based on what they've seen, and there's a lot of sharing going on the West Coast. So I've been referring to this one, and it doesn't always get all the way to species. Sometimes it just gets to the genus or the family. So that's the primary guide I've been using for the benthic species that we're seeing. It's somewhat of an in-house produced guide to help us learn more about these species.
Thank you. Jenny, I think you said it before, but I agree that there's less brittle stars in this sediment than in our prior dive in Cordell Bank.
Yeah, just an observation. There could be some underneath the sediment too.
Sure.
Seen that before. Also seeing these crazy ctenophores.
I know. Definitely our viewers are continuing to be super curious about all the ctenophores, so I doubt it would be too repetitious. Do you mind telling us a little bit more about them?
Well, they are, let me pull up some of my information about ctenophores. So we call them comb jellies. They also have some other nicknames: gooseberries, sea walnuts. These ctenophores don't have stinging cells. They have sticky cells called coloblasts. And a few species, special cilia in the mouth are used for biting gelatinous prey. So there's some that will actually swallow other jellies. I've seen that before. The Baroes comb jelly opens its mouth and will swallow another jelly. It's super cool. They don't really have they have a nerve net around their body, which helps them sense where they are and how to move in the water column.
All right, if you've been watching our dive and seen us moving along and sampling and taking lots of images, you'll see that we are kind of sitting and getting situated, and that's because we had just been conferring on a change of plans by our co-lead scientist, Jan Roletto, the research coordinator at Crater Farallone's National Marine Sanctuary, who with Guy Cochran and Sage Tezak have designed these transects to sample this area. We're kind of making some changes on the fly as we see this habitat for the very first time and decide how we best want to sample it. And it looks like we are finishing up our surveys here in this area and making a change to some other another area. So we're waiting on the navigation and the team to get set on where we're headed next.
Thanks, Danny, for the update. Maybe in the meantime, Wayne, our video engineer, can just tell us a little bit more about some of the camera work that he does to help bring such high-res images to our viewers.
Yeah, sure. What most of you are looking at on nautiluslive.org on satellite feed 1 is a HD camera that is mounted on Hercules, and it is the same camera that is mounted on Argus, which is also satellite feed 2. So those two cameras have the same resolution. While it is only 1080p or standard HD, the optics behind it are very unique and very specific for the purpose that we're using it for, which is at depth in the ocean. I have complete control over how much light enters with the iris adjustment, which I'm changing now, and I also have quite a bit of zoom available to me. I believe it's 30x zoom. And then in addition to that, I also have the manual focus control. It's a completely manual camera, and it is on Hercules. It is one of 7 cameras. There are 5 cameras on Argus. There are 7 cameras on Hercules.
Wow, I didn't realize there's 7. Neat.
Yeah, and I do not have control over the pan or the tilt. The Hercules pilot actually manipulates how the camera is pointed, and I just control the exposure and the focus and the zoom.
Yeah, thanks, Wayne.
So, yeah.
Super interesting.
All right, Danny, just an update. It'll be 4 hours at best speed.
Sounds good. We're ready to go.
All right, we're coming up tail to tail.
Okay, so we are terminating our survey at this location and just coming up into the water column a little bit and towing the ROV to the next location where you want to be. It's a little, it's quite a distance, so it's going to take us 4 hours in the water to get there.
I'm learning here that nearly all comb jellies can make light. That means bioluminescence. What we see, though, right now is not bioluminescence. What we're seeing right now is just the refraction of light from the ROVs hitting those cilia combs. That is so beautiful, a refraction of light. But in the darkness, no lights, many of these comb jellies will bioluminesce.
Some of our viewers are curious about what the most interesting discoveries so far have been in terms of Cordell Bank and the Greater Fairlawns, but also if anyone can comment on other Nautilus expeditions this year.
Well, the nav team and ROV team, they're getting all set up for this big tow. So I'd love to hear what they have, but when they're a little bit more settled. But gosh, this is my second expedition in this region, and it's just so fun to explore. It's so different every time. I think that's what I love about it. It's just, you just never know what you're going to see. And every habitat is different, and certain rocks are flowing with coral communities and some are not. And all the curiosities and wonderment of each of those for me is so fascinating. I think those skill worms today were just a cool finding that we saw earlier in the Cordell Bank Box Canyon. It's just a worm we didn't know about in this region before. There's a lot of questions about what it is, what species it is, what is it eating, what are the conditions of that habitat that are there. And I think that's, for me, the most fascinating part. One thing leads to another in terms of questions, and I love that part about science. So that's my thoughts on that. Seeing these beautiful, beautiful communities is just so stunning. Thinking about the depths and the harsh conditions of deep, deep water, and seeing all these communities thriving and interwoven it's just so cool. What about you, Danny?
Those tube worms today were my favorite thing so far on the, was that even today?
Sometime today.
I think it was at like
Yes.
4 or 5 this morning.
That's right. Kind of blends together. Because that was really, I mean, I hadn't even been thinking about that or known to look out for that. I've never seen that anywhere on the West Coast. And so that's just really neat. And, you know, the last time I was out on Nautilus was in 2017. It was the first time that we explored Cordell Bank National Marine Sanctuary, and that was just a real excitement of discovery because we just didn't know what we were going to find or if we were going to find anything. And then we found these huge bamboo corals and black corals and these really rich biological communities in the deep, deep sea. And I just remember having such wonder and excitement when we were seeing those things for the first time. So those have been some of my favorites.
And Taylor Ann, being the ocean science intern, what species are you most excited to process and learn more about?
Oh man, all of them. I can't just choose. It's all new to me. It's all really interesting. I really do like handling the corals and sponges, though. Those are pretty interesting.
How delicate, or how do you describe them out of water?
They are pretty.
I think we're having crosstalk on the line here. It's a little hard to hear. Maybe we pause on SPL. I just thought we'd pause so you can talk.
Are you guys on SPL or are you?
Sorry, yeah, Data Lab was talking and I turned you guys off.
Okay.
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 What's
so
the question?
You guys are on SPL pilots?
We didn't want to talk over you, so there was crosstalk for a while. I wanted to make sure
that's-
you got what you needed.
Yep, we are heading to a new waypoint called Greater Fairlawns 2B. which is just outside of the management area, around 500 meters depth, where we'll reacquire the seafloor.
And distance to that location?
4 nautical miles.
And at our speed, how long will it take us to get there?
Ideally, we'll be averaging 1 knot, so it'll be about 4 hours.
Okay.
We're only doing 0.4 right now, so hopefully we speed up a bit.
Okay, thank you, Miles. Thank you.
So I think this is where you play the Empire Strikes Back song? Isn't that like the beginning of the movie? Is that the right movie?
You're asking the wrong person.
I don't know. I'm not that great with the Star Wars thing, but I remember there was like a song with everything coming into focus. I always think of that with these blue water toes. Carrie, how about for you for highlights of discovery in the last 2 days?
For me, I'm just really loving comparing the differences. I mean, all just, you know, not scientifically speaking, but just the differences between Cordell Bank and the Greater Farallones. Both are two new spaces, definitely for all of us, but just seeing some subtle differences is really neat. So a specific animal, I'm forgetting what it's called, but the coral baskets with the really fine white, sometimes lightish pink, and full of the serpent stars and brittle stars. I really love those.
That's Peristinella, the one that we collected today?
Yes, correct. Yeah. That one has really captured. I'm like, oh my gosh, that is the most, you know, beautiful thing that I feel like we've seen.
Hmm.
It's really pretty. Very delicate looking.
The other thing that was really cool is there was a sponge collection today, and seeing it up close and personal, the spicules on it were just like 2 inches long. These glass spicules that stick out of the sponge, and you have to be really careful handling those because they can get in your skin and just cause so much irritation. But those were impressive.
Yeah, I had to pick it up with a large pair of tweezers and a spoon just not to get those spicules in my hands. Handled it very delicately. It was pretty heavy too.
That's an interesting thing about the collections is that sometimes you, from seeing it on the screen and making the collection underwater, and then we get them into the lab and we say, oh, that's what that was? It either looks bigger or smaller, or a lot of times it just looks smaller, but sometimes you just see features, the polyps all pull in, or realize there was tons more stuff covering it than you realized. So a lot of times things look different in the lab.
One of our viewers was curious about this next waypoint and how we kind of decided to make this change and or to not be so close to the bottom, but to get to the next waypoint by coming a little higher up.
Well, we have some large areas that we want to survey, and it takes a lot of time to bring the vehicle to the surface and then to redeploy it. So even though it's going to take us 4 hours to get there, that is actually the most expedient way to cover this ground. And so as we've talked about, we are seeing these areas for the first time, and we are making decisions on the fly about how interesting these habitats are, how well we've characterized it, and what the priorities will be by seeing, okay, we saw this on the map, and this is what it looked like actually when we ground truth it in the video, and so therefore that we might be able to make interpretations about, okay, well, if this is what that area looked like, maybe we really need to be looking over in this other area. So Jan Roletto from Greater Farallones Marine Sanctuary and Guy Cochran from USGS were making some decisions about these points, looking at the map. And I'm not sure exactly what all went into their thinking about why they wanted to make this change, but I know that those are some of the things that we're always thinking about when we're surveying new habitat.
Well, I think especially for this part of the dive in the Greater Farallon, since we're looking at some zones that are part of essential fish habitat consideration, either adding or taking away, that they really want to get to each of these areas to get a good understanding of those habitats. So to accomplish all those goals in the time that we have, we do need to scram a little bit to get to that other area that is part of that essential fish habitat consideration.
And then towing, what do we need to do to the ROVs to tow them?
I think the pilots might not be on SBL. We've actually got Antonella in her seat getting some training on from Dan, so they're chatting offline a little bit.
Antonella, we were just curious, one of our listeners was curious, what do we need to do to the ROVs to tow them?
Thank you. So we basically put them in a tail-to-tail configuration. The tether is coming out of the rear of each vehicle, so we spin them around so they're facing away from each other, and then come up and try to match the speed. So Herc can come up on its thrusters at about, right now we're doing about 14 meters per minute, and Argus, the winch can go faster than that, but we match the speed so that they're approximately at the same depth. And then we come up until we're safe. Miles is looking at the bathymetry and making sure we're not going to get towed into anything, so we come up to a safe altitude, and then we can tow the vehicles at a faster speed than we can drive along the bottom.
And it looks like we came up a couple hundred meters in the water column.
Yeah, I think we're coming up to 500 meters eventually, so it'll take a little while.
And why is 500 meters a safe depth to tow the ROVs at?
I think that's the depth of the site we're going to.
Yeah, it's a little bit shallower than our final depth. So the sooner we can get up there, the quicker we'll go, but also then we won't hit anything along the way.
That seems very important.
You know, this midwater park could be pretty exciting. There are octopuses and squid that hang out. You don't want to lose sight of the cool blue water. You just never know what's going to pop in front. And since we're transiting, we're not going to be stopping. We're booking. So it's kind of an opportunity to see this mid area, see what pops in.
I'm interested in learning more about the midwater habitat. I think that's something that we want to understand for our sanctuary, and maybe start learning more about that next. By volume, it's a huge portion of the habitat in the sanctuary, and something we still don't know a lot about. So we're looking at the seafloor. We have other projects that look at the surface, and all this area in between, we still have a lot to learn.
A lot of this layer in between is food resources, right? Diurnal migrations going up and down the water column, food web.
Yeah, there is a lot of there's a lot of biomass in this part of the ocean. So we're looking at, we'll be looking at this part of Greater Farallon's National Marine Sanctuary for the first time. And a few years ago, we were a little further south, and before the sanctuary was officially expanded, we did some surveys in the area that was to be included in the expansion area. And we made some coral collections, and Gary Williams, who's on board with us, a coral expert from California Academy of Sciences, identified that new small sea whip as Swiftia, and he named it after Greater Farallones. He called it Swiftia farallonsica. So we'll keep our eyes out for new coral species.
Saw some of that video footage. It was spectacular. Super cool. I think they saw cat shark egg cases.
That's right, yeah.
Right? All over, which is pretty neat. Seeing more reddish hues of gelatinous stuff in here.
You got Z-bias on. That's it. You're just Z-biased up. I don't know what the enable button does. No, it shouldn't. That's this. Oh, I was reading this label.
And Jenny, what are some common whale species that we find in this area?
Well, seasonally in the winter time, December, January, March, April time, gray whales pass along the coast, very closer to shore, with some beautiful viewing spots along the Sonoma coast between Sea Ranch and Walala and Point Arena. Great places to see gray whales moving through. And blue whales and humpback whales and fin whales are species that are very common to this region. They tend to follow the krill, so wherever the krill is where those whales are going to be. Humpback whales also eat fish, schooling fish, so if schooling fish are moving around, they'll follow them. There's been a lot of humpback whales near shore this year. Seen them right off the Point Reyes Lighthouse, right outside the Golden Gate, Point Bonita area. Schooling fish have been close to shore, so we've been seeing humpback whales close to shore. So those are the ones we typically see. Sometimes Rizzo's dolphins, they're also an offshore species. Dahl's porpoise, northern right whale dolphins.
Nice.
Sometimes sperm whales, but pretty hard to see. One of the toothed whales. Killer whales, orcas every once in a while, but we'll see those closer to shore. Well, not always. Sometimes you see them offshore. Near shore, offshore. So we've got quite an assortment here. There's also quite a few pinnipeds, and I'm surprised we haven't seen any northern fur seals offshore. They're a pelagic. Danny, am I getting that right? Northern fur seals? Which one is the one we see offshore?
We do sometimes see fur seals, but they're kind of not that common. More common for us to see California sea lions pretty close to shore. Sometimes we'll see fur seals. Usually just a
every
few.
once in a while.
Yeah.
And they're kind of by themselves, and they hang out offshore. So it's a treat to see them when you see them, because they come up and usually play around for a while and take off. They're really curious, but you don't see them too frequently. Other species of pinnipeds that hang out near more of the islands, the Farallon Islands, would be elephant seals, harbor seals, sometimes stellar sea lions, and sometimes we see those swimming around Cordell Bank too. A lot of animals love to be here because there's so much good food.
And Taylor Ann, someone was asking a question about what is currently going on in the wet lab. What are people working on? What will people be working on?
Yeah, so we actually wrapped up pretty much everything today from the last dive that we did, all the samples. We got them all labeled and bottled off to send to their appropriate locations and to certain scientists. So right now, the only samples that we have are the ones that we've collected today, which include a push core sediment sample, 2 coral species, and 1 Niskin water sample. So once we wrap up this dive, I'm not sure how much we'll have left to sample, if we're going to be collecting that much in these locations or not. But right now, everything was finished in the lab today. It took, what time did we start sampling, or start processing those?
I would say it took you guys probably about 6 hours or so, because we were on deck a little bit after 8. Yeah, about a little bit after 8, and then I know around like 2 o'clock, some people were still in there finishing up.
Yeah, it took a while, but it was really interesting and cool to see.
A lot of different types of samples.
Yeah, so many.
I'm always so impressed by how you guys have such a nice protocol that you follow, and there's steps where you photograph the species and then you bag them, and it just seems it's really nice to have that protocol to follow.
Yeah, good little assembly line. Gets the flow going and makes it happen a lot quicker than if it was just a one-man team. So yeah, shout out to everybody that helped and volunteered to process all of those samples and put them in their containers to be sent off to be identified.
I want to highlight an education resource that we have created on the West Coast as we have learned more about deep sea coral communities on the West Coast in partnership with our science teams. The West Coast National Marine Sanctuaries, Olympic Coast, Cordell Bank, Greater Farallons, Monterey Bay, and Channel Islands worked together to create a suite of teacher resources about the deep coral communities on the West Coast. And there's a lesson plan, there's a video, there's PowerPoint slides, and the whole lesson plan is revolving around students doing a transect. And there's videos that you can download of deep sea communities across the 5 sanctuaries, and students become the data logger and do a little transect and learn about what it's like to count species, how to identify them with a species ID guide, and what information can you get from that, and why is it important to study these areas. There's also a poster that there's an interactive poster online, and there's also one you can ask a National Marine Sanctuary to get a print copy of. And you can find this by you can just Google Sanctuaries Deep Sea Coral Curriculum. There's a link on it on the Cordell Bank website, cordellbank.noaa.gov, under Education Resources. I believe farallons.noaa.gov also has a link to it. Deep Sea Coral Communities Curriculum. And it was a nice project to put together to really highlight these unique communities that are just not really talked about much in school. And we wanted to bring a way that teachers could use it in their classroom in multiple ways, whatever works for them. It's a fantastic phenomena for teachers that are working with next generation science standards, looking at the phenomena of this habitat and the species. And I've heard myself over and over again say the questions out loud. I wonder, and I notice, and it reminds me of, and those are key questions with students in next generation science standards. I notice, I wonder, it reminds me of. They're really great questions to get students thinking about just observing what they're seeing. And I'm finding myself saying it over and over again as we're down here, which really tells me that's kind of the process of science, is just noticing first, wondering, and adds to more questions. So, little education chat. Deep sea coral communities.
We
try to
always love a little education chat. I think a cool part of Ocean Exploration Trust and EV Nautilus is a genuine valuing of community and student participation and engagement. And what speaks to that is their STEM curriculum, as well as the amazing internships and science communication fellowship opportunity that they have for EV Nautilus. So as a student, you can apply to become an intern on EV Nautilus, which Taylor Ann is, and as well as many others. And I heard earlier in the day that the applications for the internships and the application for the Science Communication Fellowship are currently open and welcoming submissions from.
I highly recommend you apply if this is something you're really passionate about and interested in. And this is a really great way to learn and to get some practice under your belt with educating others. And like Jenny said, the first step to science is just making observations and then asking those questions. And that's what I'm here doing. And logging all of those observations and keeping some of those questions in my head as I go to sleep at night.
That's great. NOAA has some opportunities too for students. We have a scholarship program for undergraduate students called the Hollings Scholarship Program, and different NOAA sites around, different offices within NOAA across the country can post projects that undergraduate students can do. And they can apply and they get training back at NOAA and then work at a NOAA office and get funded to do that internship. That's the Hollings Scholarship. We also have a graduate student scholarship called the Nancy Foster Scholarship through NOAA, National Marine Sanctuaries, and that's for master's and PhD level students. And actually, some of the collections we're doing on this trip are for one of our scholars, a Nancy Foster scholar, Karina Fish, with Bodega Marine Lab. And we've been collaborating with her to bring samples from the sanctuaries to help answer some questions that she's working on. So check out NOAA too for scholarships and internship opportunities. We have a Teacher at Sea program throughout NOAA. Teachers apply and can go to different NOAA ships. We have a Teacher at Sea that comes out on one of our at-sea monitoring programs for seabirds and mammals. And I think we'll be talking with her class tomorrow. The teacher that we had this past year.
Very cool.
Well, we're getting to some transition time. You might be hearing a little bit more chatter. We're going to call them the blue crew coming in.
Yes.
You're going to have some nice blue water.
Oh, yeah.
But I'm just going to say goodnight, and I'll see y'all at 4 in the morning if you're sticking around. And thanks for being with us.
Thanks, Jenny. Yes, this is Carrie, Science Communication Fellow. I'm going to be handing it off to Brandy, and you'll be in good hands with the next watch. So I think we told you guys, but our expeditions are divided up into 4-hour watches, and a new team of about 8 people come in every 4 hours. And they often give you a really cool new perspective. And our next watch is just filing in the control van now. So thanks everyone for tuning in. Stay with us, and we're excited about all that is to come in the Greater Fairlands National Marine Sanctuary.
For those of you that are still sticking with us on nautiluslive.org, I've just changed the views on the